Joseph. I am one of the few not computery at all people in this room, and I'm studying genetics. I'm third year, and today, this talk, I'm going to attempt to make an analogy between computer science and the basic central dogmas of genetics and molecular biology in general. Bear with me. I've been trying to do this for a couple of years now, failing miserably every time, until I started to learn a bit more of the computer sciencey stuff from mainly Isaac Mack, and an article that I found about a year and a bit ago by one of my friends. First of all, as I'm sure most of you are aware, the main thing about the genetic code is DNA. And some of you may have heard of RNA, most of you have heard of proteins. Presumably not many of you know actually what these things actually are. This is the directory that was made as a result of the Genome Project. This is open source and freely accessible. And this is the human genome. These are text files that are, each one is about 50 megabytes of a text file. And you have, okay, first of all,
ask why there's patches in human genome.
that's patch. That's just how they've labelled it, because the letters afterwards are okay. Homo sapiens. That's to do with how it's been collected. I shouldn't scroll down so much. The chromosome normally 1, or like chromosome 1, 2, 3, and these numbers refer to a particular locus, which is a particular position in the DNA. That's not what I meant to talk about. That's just to emphasize that it's big, like really big. And I'll leave that up for a minute. DNA is deoxyribonucleic acid. That's not important. It is the media by which information is stored in life. It is common throughout all living systems that we know of in the known universe so far. It is, in most organisms, read-only, equivalent to a hard. This is the hard disk. Information is stored in a digital form, however not in binary. It's actually stored in base 4, or a quaternary system. And instead of the digits 0, 1, 2, and 3, we give them the letters A, T, C, and G. These stand for chemicals, adenine, thymine, cytosine, and guanine. That's not important. So, a bit is one of these chemicals. And by the way, first of all, the double helix, which I'm not going to draw, I'm just going to draw the 2 strands of them straightened out. And you may have seen on sometimes when you've seen diagrams of DNA, you've seen the little laddery things, or more correctly, like this. These chemicals are these chemicals here and here. And they are stored within the structure of DNA itself. And the cool thing is that this backbone is stable and consistent all the way along the structure of DNA, and that's the thing that actually holds the molecule together. These chemicals are constantly moving, but they don't actually have any major effect on the integrity of the DNA. So this is the media of storage, and you can have any combination of information within the structure. And like in computer systems, how you then have a byte, which most of the time is considered 8 bits. A byte in the genetic code is 3 bits. And we call this, instead of a byte, we call it a codon. And that is 1 unit of information. What a codon does, as I'm sure some of you may be able to work out in your head, because this is a quaternary system and we've got 3 bits, to a byte, you have a total combination of not 255, but 64 possible bytes. This is of extremely high importance because what DNA does, the information that DNA actually holds, is the sequence of amino acids in protein. An amino acid is a chemical unit that has effectively a core, which is the same throughout all of them, and then a thing here. And that thing can be lots of different types of chemical structures that cause that particular thing to do something different within the body. And then you have attachment points at either side that then let you build up chains of different things, which then are generally about 3,000 or 4,000 amino acids long in an average protein. And because of the chemical interactions between these things, it spontaneously folds to produce something that may then be chemically active. And the word we give to these particular types of proteins that do things chemically are enzymes. Now, space. That's staying there because DNA is cool. In the human body, you have about 60 trillion human cells in your body. Every single cell in your body has a complete copy of the kernel, your DNA. And these, but obviously not every cell needs all of the information. For example, a liver cell doesn't need the information about being a neuron, for example. And these different sequences are if-deft out, and different combinations of comments and stuff like that that allow, in division of the cell, creation of a new process, a fork of a particular process, the dividing of a cell, allow for different sets of instructions to be present or not present during compiling. So in a liver cell can't ever become a neuron. Now, once it's been forked, it is then compiled as a liver cell, and the information is still in the source, but it's not available to the processor, if that makes sense. So you have about 60 trillion cells in your body. All of the cells have the same amount of DNA. There is about 3.2 billion bits of DNA per cell. A base is the name of one of these chemicals, A, D, C, and G, because they happen to be basic, like acids and bases, so we call them bases. Which is a bit complicated when you start getting into the measurements of these, because we use the words megabases, gigabases, and kilobases, which give the same abbreviations, KB, MB, and GB, as for information, which is a bit odd when you first start, but you quickly get used to it. And each base is about 3 nanometers across, 0.3 nanometers across. Therefore, every one of your cells has about 2.5 meters of DNA in it. Therefore, your body contains on the order of magnitude of about 1,000 astronomical units of DNA. That is 1,000 times the distance between the Earth and the Sun. Not to mention that in your body you have a lot of bacterial cells as well. In fact, you have 50 times as many bacterial cells in your body than you do human cells. They are as much a part of you as you are of them. This is pretty cool. This is why I studied this. Okay. Now, because you have these 2 strands, each of which have particular bases on them that join. You have very weak chemical bonds between these bases, but there is specificity. So, for example, this is A, this is T, then there's C, then another T, then G, blah, 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 blah. You can deduce the sequence of the other strand using what are called Watson-Crick base pairing rules, which is one of the things that they got the Nobel Prize for, which is discovering that A only ever is found opposite T. T is only ever found opposite A, C, G, T, A, and G, C. Therefore, your genome is RAID 1. It's mirrored. So if there is any discrepancy between these strands, and you don't know, and it disappears or breaks, there are mechanisms within the cells that then go, A, and then it puts a T there and fixes it and repairs. So not only is it mirrored, it automatically repairs itself. And there are very, very complicated rules and enzymes that perform these functions. They're constantly correcting any mistakes happening in your genes. Now then, when a cell divides, when a process forks itself, this is very, very heavily controlled, because forking a cell process can increase the load average on the system. It uses more resources and can slow down the time it takes for other processes to be completed and can corrupt memory. If you fork too much, if too many cells divide, or if the systems within a program, within the cell, that stop this, the U-limits and things, if you've been aware of those, that are in place to stop the overproduction, the overdivision of cells, breaks, you then get an exponential increase in load average leading to crash. This is called, in biology, cancer, leading quickly to death. Now, there are systems involved, as I've been told very recently, something called U-limits in computer science that help to limit the total load on a system by controlling the number of processes that are allowed to happen. At the end of the DNA, there are sequences called telomeres, which are, just for the hell of it, the sequence TTA-CCG. This is repeated hundreds and hundreds and hundreds of thousands of times at the ends of every strand of your DNA. And every time the cell divides, due to mechanical processes, a section of DNA is cut off. It loses a couple hundred bases at the ends. These are non-information holding sequences that are placed at the end to protect the actual information stored in the DNA. Therefore, when a cell has divided a certain amount of times, these telomeres run out and the cell dies. That is what is supposed to happen. However, when cancer cells arise, one of a very common reason why these cells have become cancerous is because they have activated the enzyme that actually generates the telomeres, because that should only ever be active during initial cell division and stem cell proliferation, which is mostly happening when you're a kid and you're actually growing, because telomeres then need to be added on to the ends of the information every time your genome divides. And cancer cells have reactivated this information, so they have the thing that extends the telomeres, thereby allowing the cell to divide an infinite number of times, which is why cancer cells are immortal and which is why you get tumors. Now, this is very, very, very similar to the halting problem that Turing invented, because just as you cannot predict if a program will ever actually end, it is impossible to create a genome or to predict if a genome or genetic system you've created can get cancer in humans. This I found out yesterday and is pretty cool. That is a perfect analogy. as far as I understand it, the halting problem is identical in genomics or in medical genetics and in computer science, which is one of the inspirations for me actually doing this talk, which I wasn't planning to do yesterday. Now, to get, if I may, how long have I gone for?
Keep on going, we don't care, it's really
Cool,
good.
fine. Anyway, thank you.
We get bored, we'll heckle.
Yeah.
Just as in, I'm going to go with things here because screw it. If you start with you make some code, yeah, and you save this code as a C file, yeah, you then, I'm not actually sure what you do, but you can do something that then makes that an object file, an O file. And then you compile that O file that then makes an executable or an exe. Is that, yeah? Okay, good. I'm only looking at Tom because he's the only one I trust. The wrong word, but yes. Screw you. Anyway, then, generally speaking, this is the only order in which these processes occur. Am I right?
Yes.
You know, generally speaking, this is the flow of your code in the structure of making the program. And it's only, you have to do wacky things to go the other way, notably reverse engineering. I never underestimate gravity. Now, in genetics, we have this thing called the central dogma, which states that DNA, which is basically the hard disk in RAID 1, because it is then transcribed. Okay, actually, this is a better way of explaining it. When the kernel, the cell, wants to call a function, it reads the DNA and puts that information into RAM. This is called RNA, which is a less stable piece of chemical, very, very, very similar to DNA, but without all the mirroring and stuff like that. And it is degraded very, very quickly. It's RAM. As soon as the cell starts doing something different, that is cleared and wiped and goes away forever. It's done in a process called transcription, which is that there. Doesn't really matter that much. I've talked about it in some of my other talks. Look at them. This is the RAM, because it is very, very, very volatile and holds only the information that the cell needs to make the thing that it wants, to do the function that it's tried to call. On a slight segue, the reason why this is necessary is mainly because DNA is too stable. You can't actually get it to do anything. The cell especially can't actually get it to do anything. As well as, the reason this is necessary is because about 97% of your DNA doesn't actually code for protein at all. It does nothing. And until very recently, it was called junk DNA. We have later discovered that a lot of it is actually involved in controlling other cellular processes and cell signaling and stuff like that. I might do another talk on that because that is another extraordinarily complicated thing that I don't actually know about yet. And this was discovered in 2005. So, yeah, not many people know about it, especially not me. And then, so the DNA contains a lot of bits that don't actually do anything that the cell can't use. Unfortunately, a lot of these things are actually within the sequences of DNA that code for a protein. And the definition of the word for a sequence of genes that code, a sequence of bases that code for a protein is a gene. And in humans and all other vertebrate type animals, because we're cool, We have things called introns. I don't know the origin of that word, but they're things that are in the middle of a gene that are not anything to do with the gene. They don't do anything and they're junk, they're bloat code. Luckily, these sections have been commented out because at the beginning of every gene, you have the base sequence UG. This does not code for it. This is one of the 64 combinations available in the base 4 system using a 3-bit byte that does not code for amino acid. This is function start. And this is actually what it's called actually in genetics. This signals the beginning of a piece that should be coded, and if this particular function is called, should be transcribed into RNA. It is then followed by whatever you want to do, and then finishes with the sequence.
Ends in another.
It ends with another codon that involves C's and G's, which is the stop codon, which then causes that particular C strand of RNA to end and to be cut off and allowed to float around in the cell. Anyway, that's really annoying. Okay. Once the RNA has been generated, once it's been called into RAM, it then does, this is where it gets a bit bleh, because you're not actually calculating anything, you're not doing anything with this data, you're then what we call translating the RNA into a protein. And that then is the molecule that actually does the stuff that the cell wants to do. That's then the thing that is chemically active and does things. You're made of protein. One of the major proteins you're made of is a protein called collagen, which is this stuff. It is everywhere. Because bodies are cool like that. It's a bit odd and really, really annoying when you're trying to grow cells because they just exhume collagen everywhere and it makes them stick to things and it means you've got to scrape them off flasks and stuff because you can't stop them making it. So if you just want the cells, they then secrete all this other stuff that makes everything stick to them, which is really annoying, but good in your cells because it's why you're waterproof and why you're not soluble, which is a good thing, I guess. Yes.
Slash wave.
Oh yes, this is something I probably should have shown a bit earlier. This is the genetic code. So these are first bases. Wait, this is weird. First base, G, A, C, and U. U is only there because in RNA there is no T. It's another chemical called U. It's just a thing that makes DNA different to RNA. It's just a thing. U is T. Sorry if this gets a bit more confusing now, but screw it, I'll carry on anyway. And you have these the second bases, and then the third bases are actually within the blocks. And each of these are the abbreviation of the name of an amino acid. They just do things. So as you can see, some things are coded by several different codons, mostly this one called arginine, which is actually coded for by I think like either 7 or 6 different codons. Whereas some, like tryptophan, which is this one here, is only coded for by 1. This is good, because tryptophan is a very, very, very reactive thing on it. And if it gets put in by accident, it can cause things to actually start doing damage. It's one of those, it's a very reactive molecule that can end up breaking things. So evolution has then led to this being only expressed with 1 codon. So if any mistake happens, it will just put in something that's not it. So the protein will work, but it won't break anything in the process. Whereas some things like arginine are coded for by loads and loads of different ones because They're ones that generally, if you want them there, that's actually to determine the shape of a protein and where it's going to go, and there are a lot more common. Also, another thing that's pretty cool is, if you see here, the things for arginine, the only difference between these is the last base. All of these are the same. And, wait a minute. So yes, so if there is a mutation, or for example, nearly every variation has the first two bases the same. Because, or the reason why this is kind of cool, is because when this process is happening, these first two bases are very hard for mistakes to be made on. The mistakes tend to happen on the last base of a codon, when DNA has been turning from DNA to RNA, or when DNA is being replicated when cells are dividing. And if one of these things are mismatched, that is the definition of a mutation. That is all the mutation means, is a change in one bit of information, potentially. So for example, the change of an A to a G mismatch in one of your proteins that is to do with moving fluid across the cell membrane is then what gives you cystic fibrosis. That is a change in one base out of 3.2 billion. That is the resolution that the DNA processing things have. I would like to see any computer match it. Because most of you don't have cystic fibrosis. Is something I've noticed here today. But, and also, that a lot of these codons, if you change one of these, it doesn't actually change what amino acid it codes for. So a lot of the common mistakes that are made, notably changing the last base, don't actually do anything. Well, here you can change the last base to any of the four, but if you want to put this amino acid here, it will always only ever put that amino acid there, which is something that evolution has done, because that's what it does. Anyway, I think I've talked for really too long. So, anyway, hope I didn't bore you. Thank you very much. Oh yeah, if anyone has any questions, feel free to ask them. And you can talk to me afterwards anytime. I will be your friend. Yes?
So in the analogy from round to protein, wouldn't it be more like the same output, dumping the round straight onto an output?
Apparently so. Okay. I know nothing about it. If it works better, then it works better. So yes, it does. What's an output then?
Just some connection to the world, rather than processing the data and doing some math with it, just simply sending it on to control something in the world.
Oh yes, that looks much better. Yes. Thank you. Any other questions?
You said quite a lot about junk bits and
Yes.
DNA. Is it really junk, or do you want just to know what it is for?
It was called junk up until people. Okay. When the Genome Project happened, which was 2001. Before it finished, everyone was really, really excited about what would happen. People were saying, we're going to cure all the diseases now, and there are going to be, out of all of these bases, because we've looked at things like, we'd sequence the genomes of things like bacteria, and bacteria don't have introns. All of their DNA does something. All of their DNA codes for things. So we figured, we're going to figure out everything to do with everything that's made in the body. And people were expecting around the level of about, 500 to about 10 million, 500,000 to a couple of million different genes, different sequences that code for proteins that actually do things. And then the Genome Project happened, and it was like, surprise, but not so much surprise. Oh, that's not what we expected. notably, instead of the 500,000 to several million genes that actually code for things that do stuff, there are about 20,000. That's a lot less than anyone ever thought there would be. And then we figured out that of these 20,000 bases, genes, they only are a couple of million bases long in total. So 97% of your DNA does not code for protein. At the time, this was thought to be the only thing DNA could do, so it was called junk DNA. And a lot of it is. For example, there are things called viruses, which are arguably living protein structures with a bit of DNA in the middle that inject DNA into organisms, into cells. And this DNA can get accidentally wrapped up in a gene, in the actual organism's DNA, and then that can be passed down, that can be inherited. So for example, we have viruses, we have viral DNA that proves that we have a common ancestor with some types of monkey and other less common vertebrates, because they also have the exact same viral insertion in exactly the same place on their genome. That hasn't changed because it doesn't do anything, so there is no evolutionary to change any of these things. But in the last literally couple of years, since about 2005, 6, they've discovered that DNA actually has a lot more control over things. So it doesn't ever make protein, it doesn't ever do anything chemically, but there are switches, there are logic functions encoded within the sequences of DNA that are involved in the switching of these processes. They're quite literally switches. There are and functions, or functions, chemical, that work in exactly the same way.
So it's a bit attached to the end of some machine, and it's basically the on-off end of the machine, and if it's on, it would be doing something.
Essentially so. It's not actually, it's not actually a change in the base sequence. We've figured out that because on the end of each, as you imagine like the DNA helix, and then the bases are within the strand, there are enzymes that add chemical groups to the outside, and these can be placed in different places, and there are enzymes that go along and read the sequence of modifications as such. And the interesting thing is that these sequences are mostly wiped when DNA replicates to make gametes, which are sex cells. So your mother's system of on-offs of switchings of gene control is wiped when you're born, but not all of it is. And therefore, this information that is added to DNA is environmental. It happens throughout your life, and it builds up due to, how much you smoke, what you eat, where you live, what temperature your body's going to be at, and blah, 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 blah. And it controls your body's reactions to a lot of these processes. But not all of these are wiped. Therefore, changes that you make in your life, whether you're obese, whether you smoke, whether you have lived in a hot country all your life and then moved to a cold one, can be inherited by your parents up to about 4 or 5 generations down the line. This has only been discovered in about the last 5 years, and this is an open question. No one has any idea how this works and why it does. This is a field called epigenetics, and it's something I really, really want to study. It's an open question. We don't know why this happens and how it does. We don't know.
Okay. I was going to ask next.
Yeah.
I recently learned that epigenetics is a thing.
Yes.
And it completely blew my mind, but I was going to ask how that fits into everything.
That is what epigenetics
so
is.
It's not, as the video I watched on it explained, it's not based on the protein DNA and what we consider the data DNA. It's this leftover, this machine logic on off the various machines in the cell.
Yes, but it's if you imagine the strand of DNA has coding sequences, non-coding sequences, blah, 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 blah. Separately to that, you then have this sequence of this other sequence that's outside, is completely unrelated to the information within the DNA at that point, as far as we're aware. Probably isn't somehow, but we haven't figured that out yet, is just another layer of information that doesn't actually, as far as we know, doesn't produce anything. It is just a switch that is then read that then controls other functions.